Psalm 119, verse 114. You are my refuge and my shield. I have put my hope in your word. In the next set of lectures on information transmission, we are going to focus on a synapse. In this first part, we're going to talk about the events that take place at the synapse. When discussing synapses, the following abbreviations will be used: NMJ, neuromuscular junction; AP, action potential; MSEC, millisecond; PSP, postsynaptic potential; EPSP, excitatory postsynaptic potential; IPSP, inhibitory postsynaptic potential; and MV, millivolts. Chemical symbols will once again include Na+, sodium; K+, potassium; Ca2+, calcium; and Cl-, chloride. The synapse is a point of communication between a neuron and another cell. That second cell could be another neuron, but it could also be a muscle cell or a gland. If that synapse is taking place between a neuron and a muscle cell, that synapse is known as the neuromuscular junction. When a synapse occurs between two neurons, the first neuron can synapse on several different places on the second neuron. If the synapse occurs on the dendrite of the second neuron, the synapse is called axodendritic. This is by far the most common type of synapse. If the first neuron instead synapses on the soma of the second neuron, then it's called axosomatic. We see this with sensory neurons. Oftentimes their central process will synapse on the soma of an interneuron in the dorsal horn. This is a bit of a faster communication because we are closer to the axon hillock of that interneuron. Remember, the axon hillock is where we have a high concentration of gated sodium channels. And then finally, if the first neuron synapses on the axon of the second neuron, the synapse is known as axoaxonic. The two most common places for this to take place would be at the axon hillock or on the axon terminal. This is the fastest means of communication and involves lots of recruitment. So typically, if we have an axoaxonic type of synapse, we'll have several of them. We see this between interneurons. The first neuron for a synapse is the presynaptic neuron. The part of that neuron that is involved in the synapse will be the axon terminal, also known as the presynaptic terminal or the synaptic terminal. Within the membrane of the axon terminal are voltage-gated calcium channels. Inside the axon terminal are synaptic vesicles. These vesicles are filled with neurotransmitter. They're made up of the same stuff as the membrane of the neuron itself. So they're a bilipid layer. They're made of fat, and they are really the same makeup as the membrane of the rest of the cell. So they're inside the axon terminal, and they're filled with neurotransmitter. They're held in place by something called synapsin that keeps them inside the terminal and doesn't allow them to migrate towards the membrane. In between the presynaptic terminal and the postsynaptic cell membrane, we have a little bit of a space, and that space is known as the synaptic cleft. And then we have our postsynaptic cell, which again could be either another neuron, could be a muscle cell, or might be a gland. On the surface of the membrane of the postsynaptic cell are receptors. Receptors are actually proteins, and they can modify their shape based upon the environment. If a neurotransmitter binds to a receptor, it will actually cause a conformational change of the receptor itself. The Let's talk about the sequence of events that take place at a synapse. The action potential propagates down the length of the axon and reaches the axon terminal. When it reaches the axon terminal, the change in voltage results in the opening of voltage-gated calcium channels. This leads to a large influx of calcium into the axon terminal. The influx of calcium triggers a series of different chemical reactions within that terminal. One of the results of some of these reactions is for synapsin to release synaptic vesicles. The freed synaptic vesicles then move to the cell membrane, bind to the membrane of the axon terminal, and release the neurotransmitter into the synaptic cleft. This release of neurotransmitter out of the synaptic vesicle into the synaptic cleft is called exocytosis. Once the neurotransmitter is released into the synaptic cleft, it diffuses across the cleft and binds to a receptor on the postsynaptic cell membrane. It takes approximately 1 millisecond for the neurotransmitter to get across the cleft and bind to that receptor. This period of time from exocytosis to binding to the postsynaptic receptor is known as synaptic delay. Synaptic delay cannot be changed. It is always going to be a constant value. I have included here a link to a YouTube video regarding synapses and the events that occur at a synapse that some of you might find helpful. Here's a still image of a synapse. So we have our axon terminal here. Within the axon terminal, we have synaptic vesicle, which is filled with neurotransmitter. In the wall of the axon terminal, we have calcium channels, voltage-gated calcium channels. When that action potential travels down the axon and reaches the axon terminal, it causes the voltage-gated calcium channels to open. Calcium rushes into the cell, and it causes the synaptic vesicles to be released where they can move down to the membrane, fuse with the membrane, and release the neurotransmitter into the synaptic cleft. The neurotransmitter then is going to diffuse across that synaptic cleft to the receptor on the postsynaptic cell membrane. When the action potential travels down into the axon terminal, causing calcium to enter the cell, this is an electrical event. Anytime you have ions moving across the membrane, it's an electrical event. But the release of a neurotransmitter, a chemical molecule, into the synaptic cleft to diffuse across and bind with a receptor would be a chemical event. That chemical event, however, is going to trigger another electrical event. Because when that neurotransmitter binds to the receptor, it opens a ligand-gated channel of some type in the postsynaptic membrane. This allows ions to move either in or out of the postsynaptic cell, creating a change in its membrane potential known as a postsynaptic potential. If this is a depolarizing change, excitatory, it is known as an excitatory postsynaptic potential, or an EPSP. On the other hand, if it's hyperpolarizing, then it would be inhibitory, known as an IPSP. An excitatory postsynaptic potential occurs when the binding of a neurotransmitter to the receptors on the postsynaptic cell membrane cause channels to open that allow ions to move, making the inside of the cell less negative. Examples would be the opening of a sodium or calcium channel, allowing for the influx of the ion into the postsynaptic cell. Each local potential is very small, only 0.2 to 0.4 millivolts. Recall that we need approximately a 15 millivolt change in order to get to threshold. So it will take approximately 50 to 100 EPSPs to reach threshold, depending upon the specific size of each individual local potential. Here's a picture of an EPSP. This is a single EPSP. So we have our stimulus, which in this case would be the neurotransmitter binding to the receptor and opening some type of a ligand-gated channel. We have an influx of sodium or calcium taking place. We get a very small local change in the membrane potential. If there's no summation, that membrane potential quickly goes back to resting. To have an IPSP, the binding of the neurotransmitter needs to cause the membrane potential to become more negative, to hyperpolarize. Examples of this would be to open up potassium-gated channel or a chloride-gated channel, which would allow potassium efflux from the postsynaptic cell or chloride influx into the postsynaptic cell. This is going to make it more difficult to generate an action potential because we're going to be moving the membrane potential further away from threshold. We're going to have a greater number of EPSPs in order to counteract the IPSP that was generated. Here's a graph of our IPSP. So we have our resting level. When the chloride enters or the potassium leaves, the membrane potential in that region gets further away from threshold. It hyperpolarizes. This is going to make it harder for us to get to that minus 55 to generate an action potential. Again, if there's no summation, we are going to quickly go back to our resting level. We have approximately 100 billion neurons in our central nervous system. Each neuron has the capacity of anywhere from 1 to 10,000 synapses. This is just an overwhelming number of possible combinations of IPSPs and EPSPs, because any of these could be occurring at the same time. It's going to become the balance of IPSPs to EPSPs, and that's going to change from moment to moment. That's going to determine if threshold is reached in any individual neuron and if the action potential then is generated or not. If we get sufficient EPSPs to end up with a net of about 50 to 100 of those EPSPs so that we get a change in membrane potential of equal to or greater than 15 millivolts, then we're going to get the action potential. If, however, the sum of our IPSPs and EPSPs is less than 15 millivolts, no action potential will be generated.